All right, welcome back. This is lecture 2, Introduction to the Immune System. The first lecture was mostly about history, and this is going to be about the different parts of your body that participate in the immune response, and then a brief introduction to the immune cells, with the next lecture being more of an in-depth discussion of the main immune cells. All right, so here's a few learning objectives. This is what I would be doing if I was teaching a class and grading it. I'd like you to be able to differentiate physical, biochemical, and cellular barriers to disease. Identify the function of primary and secondary immune organs. And be able to tell what part of your body is primary and what is secondary. And when it comes to memorizing things, I usually say to just memorize the short list first, because then anything else belongs to the other list. So for example, there's only 2 primary organs and there's a ton of secondary. So just remember the primary ones. All right. What is the composition of blood? That's pretty cool. I've got a slide that shows you, like, if you went to the hospital, got your blood drawn, and what do they do in the back room with a centrifuge? They separate it by density. And I've got a neat picture of that. And then what are progenitor cells? Those are cells before they turn into immune cells. And I'll tell you about that in just a little bit. And then where are innate and adaptive immune cells located? Briefly talked about adaptive immune cells in the first lecture, meaning B and T cells. And I'll talk a little bit more about them today. All right, my head gets in the way here. I'll try and figure it out. Okay, so why don't we get sick all the time? The answer is a lot of different things. But it starts with your skin, actually. So the fact that your skin is dead skin cells on the outside is intentional and it's clever. So your skin actually has really tightly joined cells. That's why it's called epithelial or skin cells, joined by tight junctions. Tight junctions are like, I don't know, I would almost say like screws in a piece of wood. Like the more there are, the more tightly connected the cells will be. And the whole point in having tight junctions in your skin is to prevent viruses and bacteria and fungi from accessing your bloodstream. All right. You also have your gut. Your gut includes your stomach that has a low pH. It has enzymes that break things down. Your eyes are actually really, really well protected. They have enzymes that break apart bacteria. So I always wondered if people could just take tears and put them on something and it would just kill the bacteria. I feel like if they were fresh tears, they would still have the lysozyme. Just random thoughts. Your gut also has healthy bacteria. There are 2 different kinds. There are helpful bacteria that allow you to break down your food and that stop bad bacteria from taking up residence in your body. All right, so let's talk about the physical barriers first. The physical barriers, make sure that this isn't projecting. Disappear, please. Thank you. Okay. Sorry, I'm trying to get this to go away. There, I almost got it. Yay, success, pardon me. All right, so the physical barriers restrict access to your bloodstream. I mention that because your bloodstream is one of the least well-protected areas of your body, and it travels around your entire body. So if bacteria ever gets into your bloodstream, it's really bad news. It can turn into something called septicemia, which is a life-threatening bacterial infection. Okay, so your skin is actually a little bit acidic, and that's a good thing. They also have movement inhibitors. So if your skin ever feels like a little bit oily to the touch, that's a good thing. That's actually preventing bacteria and viruses from moving all around your skin. And it helps rapidly remove infectious agents because all you have to do is wash your hands and that gets rid of a lot of the bacteria on your outer layer of skin called your epidermis. So here is the epidermis. in the dermis is protecting your blood vessels here in this reddish pink color. So it's actually very important. Okay. So there's also biochemical. These are usually enzymes, and enzymes are proteins that can do an action. All right, so enzymes are proteins that can break down essential components of an infectious agent. That's what it means when we're talking about enzymes that can protect you. Lysozyme, as I mentioned, actually comes from tears. And you might be wondering what this giant word is, peptidoglycan. If you are at all interested in microbiology, you might have heard of it. And if you're not, then you're like, what the heck is this? It is a technical chemical term for the cell wall of a bacteria. So it's more or less like poking holes in a water balloon and everything just like spills out and they die. So that's neat. Sebum. Sebum is, like I said, the kind of oily feeling on your skin. So that inhibits growth of bacteria and fungi. And then all of us actually have a unique combination of bacteria living on us, on our skin. And that is good. These are the good ones. They're called normal flora, sort of like your garden of bacteria. So they're there, and they actually know exactly how to grow on your skin, your unique type of skin, and they outcompete bad bacteria. So that's why I wrote, I'm hungry and I have nowhere to grow, because technically they will like starve out any bad bacteria that tries to land on you. Okay, so now we're switching gears from like what's kind of coating the outside of your body to what's going on inside your body. So there are two primary lymphoid organs. Remember them? Anything else is secondary. Choose the short list. So the bone marrow here in yellow is primary, and then the thymus also color-coded in yellow. Hooray for good color coding. They're your primaries. And primaries either mean where they're created or where they're educated. So bone marrow is creation and thymus is education. All right, and then the secondaries are where they go to like await their assignments. All right, so lymph nodes, I have a different slide for it, but they're located all over your body. You have like 700. Your spleen is here in blue. Looks like the lymph nodes are also color-coded blue. And then GALT is in your gut. The G is for gut-associated lymphoid tissue, also here in blue. So the secondary organ function is to help immune cells mature and encounter whatever they need to fight. All right, so let's talk about the primary ones first. We'll do bone marrow because it's the most complicated, and then we'll talk just briefly about the thymus, but I have an entire T cell lecture where we'll go in depth into the thymus. That'll be a few lectures from now. All right, so there's two types of bone marrow, and now we're gonna do some Latin. Okay, medulla osseum rubra. It's not a Harry Potter spell. A lot of this sounds like it. But rubra, hopefully that sounds a little bit like red. And osseum sounds like bone, red bone. All right, so this is the red bone marrow where your red blood cells, platelets, and most of your white blood cells are created. You have another type, osseum flava. That's yellow. Yellow is not as exciting because it's actually made up of fat. So here at the bottom, you can see at birth, all of your bone marrow is red. And as you get older, it gets converted to the yellow type. So if you're an adult like me, only around half of your bone marrow is red, which is partly why younger folks have a more robust immune system. They have more red bone marrow to refresh the immune system often. Okay. Here is another helpful hint. I have two of these because some people prefer this style of graph or whatever, table versus this style. So I'm going to talk about this one first because I think it's maybe easier visually. So part of your bone marrow has stem cells. And then as the stem cells age, it's called choose their fate. But what it means is they get more complicated. And as they get more complicated, there are less choices that they can make. All right. So the best type is called multipotent or multi-potential hematopoietic stem cell. It means it can be any cell type. And then after something happens genetically, they either choose common myeloid progenitor or common lymphoid progenitor. This is the shorter list. The two main cells that live in your lymph nodes are B and T cells. So that's part of the hint here. What cells live in your lymph nodes? So you have both T cells and B cells. And then there's also natural killer cells. So these three cell groups belong to the common lymphoid progenitor. Anything else belongs to the common myeloid progenitor. You got these giant they're literally mega karyocytes that make your platelets by breaking off little pieces. There's your red blood cells, sometimes called erythrocytes. There are mast cells, which fight parasites or cause allergy. And then there's additional allergy cells, basophils and eosinophils. And then there's neutrophils and macrophages. A lot of immunology focuses more on neutrophils and macrophages than the other cell types. Okay, but here you can see this is a really similar pathway. They just broke it up differently. So you can start off with they're calling it pluripotent instead of multipotent. Either way, it means many potentials. And then the choices are common lymphoid progenitor, which as you know now is just B cells, T cells, and natural killer cells. The other choice is common myeloid progenitor, which turns into neutrophils, eosinophils, basophils, mast cells, macrophages, and megakaryocytes. Okay. So hematopoiesis, let me try that again. Hematopoiesis is the formation of all blood cells, and it occurs in your red bone marrow. Because different places call them multipotent versus pluripotent, I just call them hematopoietic stem cells because they both agree on that. All right, so stem cells can give rise to all of our blood cells, both red and white. Now, you don't need to know like in great detail for this, but hormones and growth factors can push a cell towards one pathway or another. And once they're committed to one pathway or another, they can't change. So for example, a common lymphoid progenitor will never turn into a red blood cell. It just doesn't have the genetic programming for that. Okay. Oh, here we go. So let's say you went to the doctor's office and you had your blood drawn. It looks red. We'll talk about why. And then it actually gets spun down super fast in a centrifuge. And you get three layers. You get the red blood cells called erythrocytes. And then you get this tiny little layer of white blood cells. It's only about 1% of your whole blood. And then the rest is watery plasma. So these are like dense little iron-filled red blood cells. 45%. 1% are your white blood cells, and 55% is like the sloshy, liquidy plasma. Oh, this is a really cool scanning electron micrograph of blood. This is 18,000 times, our vision, which is incredible. So in white, you have some leukocytes, leuko meaning white cells, and erythrocytes meaning red blood cells. And then these little broken off pieces are platelets. That's what helps you clot if you ever hurt yourself and are bleeding. This is what most people would see if you worked in a lab. This is what you would see on a human blood smear, where they like take a drop of your blood, put it on a glass slide, and they like spread it out really thin. I've tried it. It's harder than it looks, but I can get it right sometimes. Okay, so there are red blood cells here, and the bigger cells are your immune cells. They're pointing out a neutrophil here, a lymphocyte, meaning a B or a T cell here, an eosinophil here, just kind of getting you used to seeing them and their different shapes. Oh, all right. So because this is like a full course that I would do, I thought I would also go over some exam style questions and then maybe even like a mock exam when I would hold them and go over some exam strategies. All right, so which of the following is not true about hematopoietic stem cell fates? Okay, so if you did remember the lymphoid, you could even write this on an exam if it helps you remember things. You can write out the common lymphoid progenitors are B cells, T cells, and natural killer cells. So then, red blood cells are derived from the myeloid progenitor. Yes. Natural killer cells are derived from the myeloid progenitor. No. B cells are derived from the lymphoid progenitor. Yes. Megakaryocytes are derived from the common myeloid. Yes. So then B is the only incorrect one. All right, so let's talk a little bit more about blood. Blood is either fluid or cells. So the fluid is called plasma, and the other parts are called formed elements. I think it makes more sense because they're cells. I showed them to you earlier. Red blood cells, white blood cells, and platelets. All right, if you ever have read some of your blood work, some of your screening, when you get back from the doctor, they might call red blood cells RBCs, and they might call your leukocytes WBCs for white blood cells. My mom was a medical technologist growing up, so she often talked about these types of things. All right, so blood is sticky, opaque, with a metallic taste. If you've ever like bitten your lip or like hurt your lip and you're like, you taste it and it's metallic because of iron. So the color varies with oxygen content. High oxygen levels are a scarlet red and low oxygen levels are a dark red. If you've ever had your blood drawn, it should be dark red because if they tried to get your blood pumping while it was scarlet red, it would like shoot out like a Wednesday Addams thing. All right, it's under too much pressure. You can only retrieve the dark red blood because it's under less pressure and less oxygen. The pH is very specific, only between 7.35 and 7.45, or we would die. Makes up about 8% of our body weight. This is not where anybody wants to go drop in weight. The average volume for males and females is only a little bit different. It's based on size, honestly. So you could be a man or a woman and have the same amount of blood if you're like the same height. Okay, so males have 5 to 6 liters and females have 4 to 5. And this is why if you ever watch crime shows, sometimes they're like, they didn't make it. And you're like, well, how do you know they didn't make it? So if you lose like 2 liters of blood, technically your body goes into shock and stops working. So if you've ever watched some crime stuff, I think you probably would know that. Okay. Red blood cells are tiny donuts. They are small diameter cells that contribute to gas transport. So they're basically here to help us with oxygen. In fact, they kick out their nucleus, which is why they don't live very long and need to be replaced constantly. So they don't even need their control center anymore. They make what they need to make, they kick it out, and they fill up with gas. So they're filled up with hemoglobin for the transport of oxygen. Okay, hang on. Okay, I'm just going to tell you a little bit about leukocytes and then the thymus. And then we'll switch gears to the secondary organs. Okay, I did mention this a little bit earlier. I actually think leuko might be Greek for white. So it means white cells. There's 2 different kinds. There's granular and agranular. I haven't showed you a really nice picture of that yet, but I've got one in the upcoming lecture that'll make this really, really clear. Some immune cells fight pathogens with granules. Some immune cells fight pathogens by eating them. So if you're going to fight with granules, you're a granulocyte. If you're going to fight with other means, including eating them, you're probably an agranulocyte. All right. They can also be talked about as living in a lymph node, a lymphocyte, or not living in a lymph node. So lymphocytes are adaptive immune cells. I already mentioned that means B and T cells. And non-lymphocytes are innate. So some of the main characteristics are coming up in 2 slides. I think I'll just go there. Oh, wait, no, here's a picture. All right, so granulocytes include neutrophils, eosinophils, and basophils. Agranulocytes include B and T cells and macrophages. So they literally have like a little grainy texture because they fight pathogens with granules filled with substances that typically do poke holes in the pathogens. Okay. And agranulocytes fight with other methods that we will go over throughout all the lectures. So here they are. I think this Venn diagram was on the end of the first lecture, but I think it's really helpful. So adaptive immune cells are slow and specific. Innate immune cells are rapid and non-specific. All right, so here you can definitely see which ones are granular or not. Macrophages are agranular, so are dendritic cells. And the rest, they all have granules, as do natural killer cells. They have granules. Okay, so adaptive immune cells, slow and specific, are B cells and T cells. These two in the middle are a bit more of like a graduate level or like an upper level course because they happen to be in the middle. Okay, so just to sum up, this one's helpful, especially if you're sort of interested in nursing or pre-med or anything like that. If you're ever forced to memorize what percent of your blood is each immune cell, it's never let monkeys eat bananas. I feel like it's one of the easier ones to remember. I've definitely heard other mnemonics that weren't as helpful. All right, so we're talking about the 1% of your blood that is made of immune cells. Most of them are neutrophils, followed by lymphocytes, which are B and T cells, followed by monocytes, which are like young, naive macrophages, and then eosinophils and basophils. So you are welcome if you ever need to memorize that. Okay. So the other primary lymphoid organ is right here, like where your neck meets, like your clavicle bones. So this is a special place for the development of T cells. It actually doesn't do anything for B cells. B cells mature by themselves in the bone marrow, so BBB, and that's a different lecture. Okay. The thymus is largest and most active during, well, childhood, so neonatal and pre-adolescent periods. By your early teens, your thymus is already beginning to shrink. That's called atrophy. So smaller amounts of T cells are generated throughout your adult life, which is like, think about the bone marrow. Our bone marrow is turning yellow and not red as we get older, and our thymus is getting smaller, which totally explains why over time our immune system is not as robust. But you can do stuff, like exercise and eating a healthy diet does tend to boost your immune system. All right, so here's the secondary ones. Secondary is where they wait, kind of like wait to mature and encounter whatever they're meant to fight. All right, so lymph nodes are bean-shaped, like literally the shape of like a pea or a bean, and that's their size too. Sometimes you can feel them swollen right here. A mosquito bit me in the back of my neck a couple of years ago, and I could feel that lymph node it's gone now, but for a couple years. Sometimes they stay large. Mosquitoes. Okay, so you have thousands. Some books say 700, some books say like around 1,000. But they're prevalent in areas of movement which is clever. They're around armpits, groin, neck, knees. It's thought that the reason light exercise when you're sick or like massage is good because it can like help move your lymph around. All right, so it contains only lymphoid cells, B and T cells. All right, so here's a cool overlap. Your cardiovascular system is in red and blue to show which blood is going like towards your heart, away from your heart, but you don't need to know that right now. This isn't an anatomy class. I just wanted to show you, like, check out where your lymph nodes overlap with your cardiovascular system. You can see how many are around the groin. They didn't draw them behind your knees, but they're there too. How many are in your armpits? Just like, oh my gosh, all over the place. That's why it's even thought that dry brushing, like if you really do like the upward motion towards your heart, potentially could also help move your lymph around. All right, so here's a lymph node. This diagram is way more complicated than I would usually want for like somebody on a second lecture. So I've actually marked out a lot of this because it's really complicated. I just wanted to point out the organization of it, and we'll come back to this. So actually everything in this pinkish color is a macrophage. I believe everything in the blue is a T cell. Yeah, mostly T cells. And then everything in yellow is mostly B cells. And this setup might not make sense right now, but it's a genius and perfect setup because the macrophages actually find and eat the pathogen. They talk to the T cells, and then the T cells activate the B cells. So it's essentially layered exactly the way you'd want like the relay race of the immune system to work. Okay. This isn't too important of a concept, but I wanted to point out that where your lymph node is can matter for how long it takes your body to figure it out. So if you got an infection here in your foot, it would need to be transported all the way to your nearest lymph node to activate your immune system. And then the immune cells have to get activated, which takes days to weeks, and then jump into your bloodstream and then travel all the way back down to your foot. So all of that takes time. Okay. Yeah. Okay, cool. So let's put some of these concepts together. Let's say that you have bacteria on your skin. Your skin gets cut. Your nice physical barrier is gone, and now the bacteria has gotten into your tissue. So they drew two immune cells here. They drew a macrophage and they drew a dendritic cell. So now they're going to fight the bacteria. They're going to eat it. They're going to break it down. And they're going to take down these pieces, parts, and they're going to enter the lymph node, the nearest lymph node. They're going to talk to the T cells. The T cells are going to talk to the B cells. And then the T and B cells are going to go back to that tissue and fight off any remaining bacteria. It's pretty cool. All right. There's a few other secondary organs to talk about. They're not as important, but I guess it depends on your perspective. So spleen filters blood rather than lymph. This is actually where like your old red blood cells go to die. So that is pretty important. One of its main functions is to bring blood into contact with lymphocytes. So this is another point where if you had an infection, your immune cells could figure it out. There's two different areas. There's the red pulp and there's the white pulp. The red pulp is macrophages and old red blood cells. That's why it's red. The white pulp is actually just B and T cells, which do appear white because they don't have any type of coloring. So this is it. The diagram's okay. I feel like it's a little bit off. I wish they had colored it a little bit better. Oh, here we go. This one's better. So the red pulp is here in this like lightish pink color. And then the white pulp is color coded blue for T cells, and yellow for B cells. So if you sliced a spleen, you would see these individual little pockets. All right. Hang in there. This is one of the last secondary organs. Sometimes it's called malt and sometimes it's called gulp. It just depends on what you're talking about. So if you're talking about any part of your body where you have mucus, then it's called MALT, mucosa-associated lymphoid tissue. If you're talking about your gut specifically, like it's a subtype of MALT. It's called gut-associated lymphoid tissue. So these are found all throughout your body, particularly like your mouth and your throat. So your throat, most of us have tonsils, but if a person would have a disorder where they lack B cells, they don't have tonsils because almost 80% of your tonsils are B cells. Your lungs are actually coated in mucosa, so are your intestines, your nasal passages, even your appendix. All right, so what is MALT? It's T cells, B cells, and macrophages. So the mucosa is incredibly important because it's one of the first places that your body encounters pathogens. So they're constantly, all of your, like, just think about it. If somebody sneezes on you, where's it going first? Nasal passage, mouth, maybe throat. So that's where it's constantly testing the antigens, meaning like the foreign proteins or pathogens that you encounter. It's also where food allergy happens. I do have a video already on food allergy. I have a pretty severe one myself and I have to carry an EpiPen. So darn it, mast cells. Okay, so gut-associated lymphoid tissue is a type of MALT. There are these little patches. They're called Peyer's patches in your intestinal tract, and they also collect antigen and check it out. Like, is this fine? Is it neutral? Is it a pathogen? So it is correctly color-coded like all of these. The yellow is B cells and the blue is T cells. So hopefully you're seeing a theme here in the way that secondary tissue is organized. Okay. So what do you think would be the worst secondary lymphoid organ to lose and why? I know I can't have you do like a pair and share unless you're watching at home with somebody. All right. So most of the time in class, people immediately say lymph nodes. And you're like, yeah, definitely. If you didn't have B and T cells, you could get the same cold. I mean, infinitely. Like you would never have a faster, better response to a cold. And boy, would that suck since there's like 800 different cold viruses. No fun at all. But if you think about it for a second, the MALT would also be really bad because the MALT is the first place where your immune cells encounter a pathogen. So it's like the alarm bell goes off probably in the MALT first, most of the time. So you might be waiting days. And in those days, the virus and the bacteria or the fungi are having all that time to like be in stealth mode and just make copies and copies of themselves and wreck your tissue until finally a lymph node would figure it out later on, like days later. But the MALT could figure it out right away because it's typically where you first encounter things. So yeah, the students often I have them debate whether or not never remembering something versus like always having to wait days before you can get like a start on fighting the pathogen. And the spleen, I mean, honestly, people do okay without their spleen. So sorry, spleen. All right, last two slides. This is just to kind of put together what's happening over time with the innate and adaptive immune system. So stage 1, some kind of infection happens. Ideally, this is a really short stage. Ideally, it's identified right away by your innate immune cells. The innate immune cells travel to a lymph node and induce the adaptive immune response, meaning they tell the T cells, and the T cells tell the B cells, and they both go back and fight off the infection together with other innate immune cells. Sometimes it can take 2 weeks for T cells and B cells to get fully activated and make enough copies of themselves and get back to where they need to go. But step 4 is the cool step. That is immunological memory. So some percentage, a small percentage of your B and T cells retain the memory forever. So the next time you would get this infection, this blue arrow would be much, much, much shorter. So you could fight back in about 3 days instead of a couple of weeks. All right, we made it. Immune cells are involved in multiple organ systems. Hopefully you can name a few. The innate immune system informs the adaptive immune system, which promotes a response. The primary immune organs are where they're created or educated, and secondary are where they mature and become activated. Blood is mostly composed of the liquid plasma, red blood cells, and white blood cells, but only 1% white blood cells. They still get the job done most of the time. Innate immune cells are in the bloodstream. Adaptive immune cells are mainly in your lymphoid organs. White blood cells can either be granular or agranular. I will expand on that in the next lecture. And then hopefully you can remember never let monkeys eat bananas, and that there's only 3 common lymphoid progenitors: B cells and T cells, and natural killer cells. So hematopoiesis is the formation of all blood cells. All right, thanks for tuning in, and I'll have another lecture up soon.